Hello, I'm Essay, your AI essayist. Please note that all content, audio and video in this presentation are AI generated. As AI can sometimes produce inaccuracies, I encourage you to verify any factual information presented. With that in mind, let's begin.
Here is the generated essay. The year 1962 witnessed the publication of a monumental work that reshaped the landscape of biological understanding, the molecular biology of the gene by James Watson. This treatise served as a cornerstone in establishing molecular biology as a distinct and rigorous discipline, synthesizing burgeoning knowledge of genetics, biochemistry, and structural biology into a cohesive framework. This essay undertakes a comprehensive evaluation of its claims, arguments, and specific details, section by section, to assess its enduring significance and internal consistency. It presupposes a readership familiar with the scientific terminology and intellectual currents of the period. Section 1. The genetic material claims of DNA's primacy. The opening chapters of the molecular biology of the gene address the fundamental question of heredity's physical basis, unequivocally asserting deoxyribonucleic acid, DNA, as the primary genetic material. Watson meticulously elucidates the experimental evidence supporting this assertion, drawing heavily on experiments conducted by Avery, MacLeod, and McCarty, who demonstrated the transforming principle in bacteria was deoxyribonucleic acid. He expounds on the subsequent work of Hershey and Chase, whose bacteriophage experiments provided compelling evidence that DNA, rather than protein, carries the genetic information during viral infection. A central claim elaborated in these sections regards the inherent stability and information-carrying capacity of the deoxyribonucleic acid molecule. The helical structure, initially elucidated by Watson himself along with Crick, posits a mechanism for faithful replication and preservation of genetic information across generations. Detailed structural explanations are provided, accompanied by schematic diagrams that illuminated the arrangement of nucleotides, the deoxyribose phosphate backbone, and the hydrogen bonding that unites the two strands. The presentation here avoids equivocations, presenting the available evidence with a confidence bordering on conviction. The argumentation persuasively integrates findings from seemingly disparate experiments to build a cogent case for deoxyribonucleic acid as the primary bearer of heredity. It should be noted that Watson acknowledges the lingering uncertainties surrounding minor bases in deoxyribonucleic acid and their prospective roles in fine-tuning cellular processes, a prescient observation given subsequent discoveries of epigenetic modifications. Section 2. Deoxyribonucleic acid replication, the semi-conservative model. The book dedicates significant attention to the mechanism of deoxyribonucleic acid replication, articulating the semi-conservative model proposed by Watson and Crick, and substantiated experimentally through the elegant experiments of Meselson and Stahl. The central claim here centers on the idea that during replication, each strand of the parental deoxyribonucleic acid molecule serves as a template for the synthesis of a new complementary strand, resulting in two daughter molecules, each containing one parental strand and one newly synthesized strand. The argument is bolstered by a careful exposition of Meselson and Stahl's density gradient centrifugation experiments, utilizing isotopes of nitrogen to distinguish between parental and progeny deoxyribonucleic acid molecules. Watson meticulously explains the experimental design, the observed banding patterns in the centrifuge tubes, and the logical deductions that support the semi-conservative model. He emphasizes the conceptual elegance of the semi-conservative mechanism, highlighting its efficiency in maintaining genetic fidelity. The discussion extends to the enzymatic machinery implicated in deoxyribonucleic acid replication, introducing deoxyribonucleic acid polymerase and describing its role in catalyzing the addition of nucleotides to the growing deoxyribonucleic acid chain. The section further considers challenges associated with deoxyribonucleic acid replication, such as the need for an initiation point and the inherent difficulty in replicating the ends of linear chromosomes, the chapter presents a lucid and compelling account of the semi-conservative replication mechanism, solidifying its place as a central dogma in molecular biology. At this point, Watson acknowledges the ongoing search for specific molecules involved in replication, initiation, and termination, highlighting the limitations of existing knowledge and stimulating further risk. Section 3. Gene structure from cistron to operon. Watson next proceeds to dissect the organizational architecture of genes. He introduces the concept of the cistron, a functional unit of heredity encoding a single polypeptide chain, thereby connecting deoxyribonucleic acid sequences to protein products. This section grapples with the intricate organization of genes within acterial chromosomes, laying specific focus on the operon model as proposed by Jacob and Monod. The central claim focuses on the clustered arrangement of genes involved in a related metabolic pathway controlled by a single regulatory element. The lactose operon in Escherichia coli, E. coli, serves as the primary illustrative example. Watson meticulously details the components of the lactose operon, including the structural genes, the promoter region, the operator region, and the regulator gene. He explains the mechanism of induction, whereby the presence of lactose triggers the expression of the structural genes through an activation of the repressor protein. Watson emphasizes the elegance and efficiency of the operon model, allowing coordinated expression of genes involved in a specific metabolic pathway. He acknowledges potential complexities in its application to eukaryotic systems, where gene organization and regulation are considerably more intricate. Specifically, the discussion of gene structure and the cistron concept leads to a consideration of the relationship between deoxyribonucleic acid sequence and protein sequence. Watson articulates the concept of the genetic code, emphasizing the triplet nature of codons and the degeneracy of the code. He further explains how the code was deciphered through a combination of biochemical experiments and genetic analyses. Section 4. Ribonucleic acid, the messenger, and more. The discussion turns to ribonucleic acid, or ribonucleic acid, emphasizing its crucial role in mediating the flow of genetic information from deoxyribonucleic acid to protein synthesis. Watson outlines the different types of ribonucleic acid, including messenger ribonucleic acid, transfer ribonucleic acid, and ribosomal ribonucleic acid, each fulfilling distinct functions in the processes of transcription and translation. The central claim here is concerned with the messenger ribonucleic acid molecule, or messenger ribonucleic acid molecule, acting as an intermediary carrier of genetic information from the deoxyribonucleic acid template in the nucleus to the ribosomes in the cytoplasm. The process of transcription, in which the deoxyribonucleic acid sequence is copied into a complementary ribonucleic acid sequence, is comprehensively described. Watson explains the role of ribonucleic acid polymerase in catalyzing the synthesis of messenger ribonucleic acid molecule, highlighting the similarities and differences between deoxyribonucleic acid polymerase and ribonucleic acid polymerase. The subsequent process of translation, in which the messenger ribonucleic acid sequence is decoded to synthesize a polypeptide chain, is elucidated with meticulous detail. Watson describes the role of transfer ribonucleic acid molecules in carrying specific amino acids to the ribosome and matching them to the appropriate codons on the messenger ribonucleic acid. The structure of the ribosome, composed of ribosomal ribonucleic acid and proteins, is characterized. The discussion highlights the ribosome's role as the site of protein synthesis, catalyzing the formation of peptide bonds between amino acids. The explanation addresses the crucial functions served by each form of ribonucleic acid, highlighting both their unique contributions and their coordinated efforts. Section 5. Protein synthesis from ribosomes to polypeptides. A focused discussion of protein synthesis follows, elaborating on the mechanisms by which genetic information encoded in messenger ribonucleic acid is translated into polypeptide chains. The central claims focus on the intricacies of the translational process, encompassing multiple stages: initiation, elongation, and termination. Watson describes initiation by small ribosomal subunits binding to messenger ribonucleic acid, recognizing a start codon, typically adenine, uracil, guanine. Transfer ribonucleic acid carrying methionine then pairs with the initiation codon, forming initial translational complex. Then, elongation begins when the next transfer ribonucleic acid molecule corresponding to the following codon binds to the ribosome's acceptor site. A peptide bond forms between adjacent amino acids, catalyzed by peptidyl transferase, and the ribosome translocates along the messenger ribonucleic acid, moving one codon. The discharged transfer ribonucleic acid leaves, making room for the next, repeating elongation until the ribosome reaches a stop codon: uracil, adenine, adenine, uracil, guanine, adenine or uracil, adenine, guanine. These codons signal termination. As there is no transfer ribonucleic acid matching them, release factors bind, causing the polypeptide chain to detach and the ribosome disassembles. Watson provides a detailed account of the enzymatic factors involved in each stage of translation, emphasizing the complexity and precision of the process. He acknowledges ongoing debate regarding the precise mechanisms of ribosome translocation and the regulation of translational efficiency. He provides insight into processes such as messenger ribonucleic acid editing, where alterations to sequence occur post-transcriptionally, highlighting the dynamic nature of genetic information. Section 6, Regulation of Gene Expression Control Mechanisms. Watson next delves into the mechanisms governing gene expression, emphasizing the control mechanisms that dictate when and where specific genes are activated or repressed. Although the operant model provides a well-defined example of gene regulation in bacteria, Watson broadens scope to encompass other regulatory mechanisms and complexities, conceding that much remains unknown. The central claim focuses on the adaptive nature of gene expression, allowing cells to respond to environmental stimuli and developmental cues. Watson discusses the role of regulatory proteins, such as activators and repressors, in modulating gene transcription. Activators bind to deoxyribonucleic acid sequences near protoregions, enhancing transcription initiation, while repressors bind to operator regions, blocking ribonucleic acid polymerase from accessing the promoter. He addresses the phenomenon of catabolite repression, where the presence of a preferred energy source, such as glucose, inhibits the expression of genes involved in the metabolism of alternative energy sources, such as lactose. He also recognizes the importance of chromatin structure in regulating gene expression in eukaryotes, noting that tightly packed chromatin is generally inaccessible to transcription factors, while loosely packed chromatin is more readily transcribed. He mentions post-translational modifications of histone proteins, such as acetylation and methylation, in influencing chromatin structure and gene expression, observations prescient of the later field of epigenetics. Section 7, Mutation and Recombination. Alterations to the genetic code. The text shifts focus to explore the processes of mutation and recombination, which introduce variations into the genetic code. Watson discusses the different types of mutations, ranging from point mutations, involving single nucleotide changes, to frameshift mutations, involving insertions or deletions of nucleotides. The central claim focuses on the impact of mutations on protein function, ranging from silent mutations that have no effect on protein structure to nonsense mutations that result in premature termination of translation. Watson explains how mutations can arise spontaneously due to errors in deoxyribonucleic acid replication, or they can be induced by exposure to mutagenic agents, such as radiation or chemicals. He illustrates the process of deoxyribonucleic acid repair, where cellular mechanisms identify and correct damaged or mismatched nucleotides, maintaining the integrity of the genome. Additionally, Watson explores the process of recombination, in which genetic material is exchanged between homologous chromosomes, generating novel combinations of genes. The chapter elucidates the importance of recombination in generating genetic diversity, which is essential for adaptation and evolution. Watson details that recombination occurs via breakage and reunion of deoxyribonucleic acid double helixes, catalyzed by specific enzymes. Genetic analysis uses recombination frequencies to map genes, allowing visualization of genetic relationships. Section 8 Extrachromosomal inheritance, plasmids and viruses. Watson broadens his scope to consider extrachromosomal inheritance, focusing on the role of plasmids and viruses in transmitting genetic information independently of the main cellular chromosome. The central claim is centered on these elements contributing to genetic diversity and adaptation, particularly in bacteria. Plasmids, small circular deoxyribonucleic acid molecules separate from the chromosome, often carry genes conferring antibiotic resistance or other selective advantages. Watson explains how plasmids can be transferred horizontally between bacteria through conjugation, transduction, or transformation, facilitating the rapid spread of antibiotic resistance genes. He explains the life cycle of bacterial viruses, or bacteriophages, describing their ability to inject deoxyribonucleic acid into host bacteria, hijacking cellular machinery for replication. He discusses lysogeny, where bacteriophage integrates into the host chromosome, replicating along with it, and how environmental triggers can activate lytic cycle, resulting in cell lysis and the release of many new virus particles. Watson also touches on the properties of animal viruses, including their diversity in genomic structure and replication strategies. He acknowledges the potential for viral integration into host genomes and its implications for cellular transformation and oncogenesis. The chapter serves as a reminder that genetic information is not strictly confined to the cellular chromosome, but can also be transmitted through extrachromosomal elements, influencing cellular phenotype and evolution. Section 9 Immunological systems, a glimpse of complexity. Moving beyond the core themes of molecular genetics, Watson includes a section touching upon immunological systems, showcasing the complexity of biological systems and highlighting the molecular basis of immune responses. The central claim describes the specificity of antibody-antigen interactions, enabling the immune system to recognize and neutralize foreign invaders. Watson outlines the structure of antibody molecules, composed of heavy and light chains joined by disulfide bonds. He discusses the immense diversity of antibodies generated through combinatorial joining of gene segments, somatic hypermutation, and junctional diversity. He touches on the role of T cells in cell-mediated immunity, recognizing antigen fragments presented on cell surfaces by major histocompatibility complex molecules. He briefly discusses immune tolerance, the mechanisms preventing immune system from attacking self-antigens. The inclusion of immunology, while somewhat tangential to the central themes of molecular genetics, underscores the broader implications of molecular biology for understanding complex biological phenomena. It offers glimpse into the molecular basis of immune responses, highlighting the elegance and sophistication of the immune system. Section 10 Some future directions, speculations and predictions. In the concluding section, Watson speculates on future directions in molecular biology, outlining the potential for applying molecular insights to address pressing biological and medical challenges. The central claim is rooted in the transformative potential of molecular biology for understanding and manipulating life processes. Watson predicted advancements in gene therapy, offering the prospect of correcting genetic defects and preventing disease. He foresaw the development of new diagnostic tools based on molecular analysis, enabling early detection and personalized treatment of diseases. Watson also considered the ethical implications of molecular biology and the need for careful consideration of the societal consequences of new technologies. He discussed the potential for genetic engineering to improve crop yields and enhance human health, while also acknowledging the risks associated with altering the genetic makeup of organisms. The concluding section serves as a testament to the transformative power of molecular biology and its potential to reshape our understanding of life and medicine. In summation, James Watson's The Molecular Biology of the Gene stands as a landmark achievement in scientific literature. Through meticulous exposition, clear argumentation, and insightful synthesis of experimental evidence, Watson provided a comprehensive overview of the burgeoning field of molecular biology. His systematic evaluation of genetic material, deoxyribonucleic acid replication, gene structure, ribonucleic acid function, protein synthesis, gene regulation, mutation, recombination, extrachromosomal inheritance, and immunology laid the groundwork for immense future progress, though specific details have been revised and expanded upon by subsequent research. The foundational principles articulated in this text remain cornerstones of modern biological understanding. The book's legacy extends. Beyond its scientific content, it served as a catalyst for widespread adoption of the molecular approach across a diverse range of biological disciplines, solidifying molecular biology as a central and indispensable pillar of the life sciences.
We really appreciate you tuning into this AI-powered presentation. Our goal is to push the boundaries of AI in content creation, and your input is invaluable. If there's a particular subject you'd like to see us tackle next, or if you have suggestions on how we could fine-tune our AI's creative prompts, please don't hesitate to leave your ideas in the comments section. We're excited to bring you more unique content very soon.